stuck in the microphone. Okay, so today I'm going to give you a talk about some of the work that's been going on in the lab for a while. The title is Convergence at the Level of the Phenotype Mirrored Across the Genome? The Repeated Evolution of Glyphosate Resistance in the Common Morning Glory, Ipomoea, Purpurea. And I want to say this is funded by USDA as well as University of Michigan. And this is the work of Dr. Megan Van Etten, a previous postdoc in my lab who is now at the University of Pennsylvania. So phenotypic convergence is this idea that the same phenotype evolves across multiple species or separate populations of the same species due to exposure to the same environmental stressors, environmental agents. So you have separate lineages evolving a similar phenotype. The idea has been present in the literature since Darwin, where he said, for animals belonging to two most distinct lines of descent may readily become adapted to similar conditions and thus assume a close external resemblance. So we have a lot of examples of this, which I might show you, and I might not. I think this died. In nature, where we have the repeated evolution of, say, metal tolerance across separate lineages or separate species of plants, the repeated evolution of herbicide resistance, TDX resistance from our garter snake example, and a lot of different examples in the literature now about the evolution of coat color and body color, as well as floral color. So we often have the same phenotypes arising among very distantly related species. And what a lot of these systems have in common, or a question that they often ask in common, is convergent phenotypic evolution the result of parallel genetic changes among lineages or species? So is the same genetic basis responsible for these same phenotypes across divergent lineages or species? And so in the case of herbicide resistance, I'm going to give you a very restricted case here. I'm showing you the causes of convergence at the phenotypic level. So panels A and B up there are showing you the convergent evolution of herbicide resistance across separate species. It's due to the same physiological mechanism in both species, but it's due in panel A to the same mutation in the same locus, whereas in panel B we have a different mutation in the same locus responsible for resistance. In panel C, I'm showing you a different situation where you have the convergent evolution of herbicide resistance across species, but it's due to different physiological mechanism across these species and different loci or different genes at the genetic level. So the first two examples that I'm giving you, it's really an example. It's also been called extreme parallelism or hotspots of gene reuse. So this is the situation where there's a very limited genomic landscape that can respond to, say, some kind of a stressor. And this idea has been buffered by many studies of pesticide resistance evolution. So whether it's insecticide resistance or herbicide resistance or fungicide resistance evolution, a lot of examples come from the xenobiotic literature showing that the same genes are responsible for the resistance phenotype, and often the same mutations in the same genes are responsible for the resistance phenotype. So much so that Martin and Orgozo appearances of insecticide resistance highlight a restricted adaptive landscape of accessible mutations. There's just not that much genomic space that could respond to this very strange stressor, especially in the case of xenobiotics. And so I'm interested in this question, is the repeated evolution of resistance across the landscape due to similar or different changes at the genetic level? And to answer this question, we're using the common morning glory, Ipomoea purpurea, which is a noxious weed found mostly in agriculture, soy, cotton, and corn crops. It is evolving resistance to glyphosate, which is the active ingredient in the herbicide Roundup. I'm sure most of you are probably familiar with Roundup. So we've done some studies in the lab to assess the level of resistance in nature in the common morning glory. So we did a large dose response curve where we went out to about 47 different populations. We collected seed. We took them to the greenhouse, grew them up, sprayed them with about 6 different levels of glyphosate. I'm showing you here the results from 2 times the field dose of herbicide, and what I'm showing you here is the proportion of individuals that survived from each population. So the populations that are red tended to have high resistance. A lot of individuals survived the application of herbicide. The populations in blue tended to be highly susceptible. So what you can see from this figure is that we've got two areas of the southeast where we have high levels of resistance. So we have one center of resistance in central Tennessee, as well as one center of resistance in the coastal plain of North Carolina. So we wanted to know, are the same genes, are the same mechanisms responsible for herbicide resistance across these very separate populations? And the first thing that we decided to do was to sequence up the target locus of Roundup, which is the EPSP synthase locus. It's a gene that controls the protein that interacts with Roundup in the shikimate acid pathway. And it's the first thing that generally weed scientists do. We know how herbicides are interacting with target proteins in plants, so we're going to go and sequence up that locus and see if we find any mutants associated with resistance. And so that's what we did. We took about, I think it was 5 different individuals from 8 different populations, and we sequenced up the EPSP synthase locus across these individuals, but we found no evidence for variation that's associated with resistance across these populations. There's no indication that there's a change at the genetic level within the target locus of Roundup leading to resistance, which that means that it can be anything else. It could be any other mechanism. Maybe the herbicide is not getting into the plant. Maybe the herbicide is being detoxified or not translocating throughout the plant. So now they really had a blank slate to try and figure out what's causing resistance in this species. So the next thing we decided to do was a population genomics approach to address our question. So we actually have 2 questions here. What's the genetic basis of resistance across these populations? And is it the same genetic basis across these separate populations? So we did population sampling where we took 4 susceptible populations, meaning that they showed less than 20% survival, 4 highly resistant populations, so greater than 70% survival, 10 individuals per population. And we started by doing some next-rad sequencing, which is a genotype by sequencing protocol. We amplified the selective sequence to look for SNPs. From this, we identified 8,100 SNPs. So now we have 8,100 randomly sequenced SNPs. And we wanted to identify and see if there's any signs of selection on these loci that might indicate that they are responding to selection from herbicide. So we did this running 2 different programs to look for signatures of selection. We ran Bayscan, which basically looks for FSD outliers or high FSD structure of loci. We also ran Bayn, which is a program that looks for correlations between allele frequencies and an environment. In this case, our environment was the level of resistance. So we ran these 2 programs on our 8,100 loci. Bayscan identified 47 loci that appear to be under selection. Bayn identified about 83 loci that appear to be under selection. So we had around a bit over 100 loci that seemed to be showing signatures of selection across our resistant populations. Now that's not really great data for saying this is the genetic basis of resistance, right? These are just loci that are showing signs of selection, and there could be other forces that are leading to patterns like that. So we decided to increase our precision a bit by doing target capture resequencing. We took our loci that showed signs of selection, we aligned them to an assembly of the Ipomoea purpurea genome, and then we looked around those SNPs for loci of interest. We designed probes of exons around those genes that had an outlier SNP. So we designed probes for exons for about 200 of them that were outlier genes and 200 that were control genes. And we did some targeted resequencing. We resequenced 5 individuals per of our 8 populations, and then looked for signs of selection by comparing our resistant and susceptible populations. So just to give you some stats from this resequencing, we had about 11 million reads per individual. We were able to assemble 67,000 contigs. This led us to 152,000 high-quality SNPs on 27,000 contigs. Before we did our screens for selection, we reduced it to about 1 SNP per every KB. So we're assessing signs of selection, again, on our resequenced data. We again used the programs Bayscan and Bayn, and this pulled us back a bit over 200 loci, again, that are showing signs of selection. Okay, so we've resequenced these areas of the genome that seem to be showing signs of selection. We now have more complete assemblies of area around those loci that are showing signs of selection. So what do we do next? We decided to align them to the genome of a close relative, Ipomoea nil. And I'm showing you here the chromosomes of Ipomoea nil 1 through 15. We found that 71% of our outliers from our resequencing data tended to occur within 5 regions of the genome. And so I've shaded those there for you, chromosomes 1, 6, 10, 13, and 15. These areas are all different sizes, and to get the sizes, we just kind of eyeballed it, quite honestly. Okay, so we found these regions that seem to have a lot of outliers within them. What's in the region? So now we've decided to go in and look for genes of interest within these regions that are showing signs of selection. We find evidence for ABC transporters, glycosyltransferases, GSTs, and cytochrome P450s. And not only do we find evidence of these genes, we also find multiple copies of these genes within these regions. Now, what are these genes? What do they do? They're part of a hypothesized herbicide detoxification pathway. In phase 1, you have your cytochrome P450s that oxygenate the herbicide molecule and allow it to be noticed. And then in phase 2 is conjugation, and GSTs and glycosyltransferases will add sugar molecules to the molecule. And then in phase 3, that's transport and degradation, and ABC transporters are involved in transport and degradation. So we're finding genes in areas of the genome that are under selection, and these genes have things to do with herbicide detoxification. Okay? So I've mentioned that we found multiple copies of these genes from these gene families. Well, because they're from gene families, we wanted to know, are we finding them just due to chance, because you find them often in plant genomes because they're from gene families? So we decided to address this by doing some resampling. We resampled the Ipomoea nil genome and came up with an expected number of genes from each of these families. You can see that for glycosyltransferases, ABC transporters, and cytochrome P450s. There's our expected number of genes from the genome. There's our observed number of genes. So we're finding more often than not genes from these gene families than you would expect by chance. Okay, so we have an idea from this broad screen that the species might be detoxifying glyphosate. We know that it's not a target site locus. We know that EPSP synthase locus isn't doing anything to the plant. We think there's some herbicide detoxification going on. The next question that we wanted to address is, are the populations doing it in the same way? Okay, so is it the same mechanism across these different populations? And we have some evidence that that might be the case, at least from chromosome 10. So the area on chromosome 10 showed reduced diversity. Okay, so I'm showing you the nucleotide diversity of these regions that had those outliers. And you can see in red, we've got the resistant individuals. In blue, we've got the susceptible individuals. And we've done it for chromosome 1, 6, 10, 13, 15, et cetera. The only chromosome that showed any pattern of reduced variation in the resistant individuals compared to the susceptible individuals was from chromosome 10. Okay, so what's going on in chromosome 10? In chromosome 10, there are 7 duplicated glycosyltransferase genes, as well as 9 cytochrome P450 genes. Let's take a closer look at a 42 kb segment from this region on chromosome 10. I'm showing you the 7 duplicated glycosyltransferase genes right in a row. And I think it's the 5th glycosyltransferase gene had evidence for very reduced nucleotide diversity in the resistant individuals compared to the susceptible individuals, suggesting that something is going on with this particular glycosyltransferase that's related to resistance. And it's the same across all resistant populations. The other thing I want to point out from this figure is that the resistant individuals have a very different haplotype than the susceptible individuals, regardless of what population they're from. Okay, so to conclude, we find that resistance is not due to changes in the EPSP synthase locus. We're not talking about a target site mechanism with this species. We have at least 5 regions of the genome that respond to selection for resistance in the common morning glory. We think we have a potential detoxification mechanism. And we have what we think is a parallel genomic response, at least on chromosome 10. And I didn't show this data because I was worried about time, but we also have evidence for selection on standing variation within these populations or migration between the populations. We have evidence for very high selection in this region on chromosome 10, and that it's from a low frequency, an initially low frequency variant. Okay, so with that, I would like to thank people from my lab, past and present, and again, the funding from USDA and University of Michigan, and again, acknowledge Megan Van Etten's work. With that, thank you.